essentially, I want to go over some of the basics and fundamentals of the immune system. And the audio lecture on Wednesday will actually be going over the histology of the major immune system organs and structures. For instance, we'll be looking at the diffuse lymphoid tissue, the lymph nodules, the lymph nodes, the thymus, the spleen, and the tonsils. And so we'll be actually looking at those in more detail in some of their characteristics. But today's a little more of a background. Some of it, at least I hope, will be reviewed, but there may be some new things as well. Decide to kind of get us oriented to the immune system and what it's like. First of all, when we look at the immune system, which is essentially the body defense system, what we're trying to accomplish is to protect the body from organisms that can cause disease, make us sick. That's the main purpose of the immune system. Now, the immune system does other things. For instance, if you do an organ transplant or a blood transfusion that doesn't match, then you also have your immune system, which is attacking those transplanted cells. You can also get autoimmune diseases where your immune system inappropriately attacks your normal cells and causes a problem. And there's a number of diseases that are autoimmune. We can also have a over-responsiveness to things that aren't really pathogens. And this can cause allergies and even dangerous overreactions by the immune system, anaphylactic shock, which can be deadly. And so there's a number of things that the immune system maybe reacts in somewhat inappropriate ways. But its main function is to fight off pathogenic or disease-causing organisms and problems. This would include pathogenic bacteria, viruses, and parasites, worms, things like that. So how's it going to do that? Well, first of all, the first order of defense, if you will, is to try to keep the pathogen out of the body. And our skin does that. We'll be looking at the skin later on this week, but the skin is a protective barrier that prevents a lot of things from getting into our internal environment. If we have damage to the skin, like if we cut through the skin or we have burns or whatever, it makes it easier for pathogenic agents to enter and cause problems. We also have enzymes, which are antibacterial, antipathogenic. Lysosome is one. It's in the tears, it's in the saliva, and that helps protect us. Acid, there's a little bit of a lower pH on the surface of the skin, a little acidic environment, and then, for instance, in the vagina and the stomach, that helps prevent pathogenic organisms from taking hold. In the respiratory system, we have mucus with cilia that keeps pushing dangerous pathogenic organisms away from the lungs. So these are all something somewhat helpful for us, very helpful for us to try to protect ourselves. Now, when we look at this diagram, what we're seeing here is some of the components of the immune system. And we'll look at some of these briefly here and then in more detail eventually. One of the things that we see is whenever we have a duct or tract that opens to the exterior, it is a point of danger for the body. This would include the respiratory system, the digestive system, the reproductive system, the urinary system, which is similar. And so when we have these tracts or ducts the lining wall of these tracts or ducts is called a mucosa. And so these lining mucosa really are an epithelium and an underlining connective tissue. So all the way along our digestive tract, we have an epithelium and an underlying connective tissue. And that's true for the respiratory system, and that's true for the urinary system. And so this mucosa, because it's exposed to the external environment, remember, everything inside the digestive tract and the respiratory tract and the urinary tract is in the external environment, that lumen, because it is exposed to the exterior. And so that is a point of danger. So we need to have body defenses guarding those tracts, those linings. And so we have what is called MALT, mucosa associated lymphoid tissue. This is going to be like diffuse lymphoid tissue and lymph nodules that we'll get into some detail on Wednesday lecture, whenever you listen to it. Specifically, if it's in the respiratory tract lining, it's called BALT. That stands for bronchial associated lymphoid tissue. If it's along the digestive tract, it's called GALT, which is gut associated lymphoid tissue. Interesting name. And so this helps guard the entrance, helps prevent pathogens from getting on into our internal environment. Then we have an entire system called the lymphatic system, shown in green here. The lymphatic system has a number of one-way lymph vessels. And what these do is, number one, they collect the excess fluid from our tissue or interstitial fluid. And it takes it on a slow, meandering journey back and finally dumps it into the regular circulatory system, into the venous system. And so it makes its way back, and it dumps it back finally into the thoracic duct. This is where the lymph fluid dumps back into the regular circulatory system. But on its way, from all through the body, this lymph fluid is moving through a number of lymph nodes. Lymph nodes are organs. They have a capsule around them, and they have lots of cells that are going to be part of our immune system and are going to attack any potential pathogen. They need to recognize it and attack it. And so that's what they do. And this is why sometimes when we get sick, we can even feel swollen lymph nodes, like in our neck area. You might feel, oh yeah, your lymph nodes are a little swollen. Well, we have patches of lymph nodes throughout the body, in the groin area, in the axillary underneath the arm, in the neck, in the abdomen, lots of places. And the lymph fluid goes through these lymph nodes. And we're going to look histologically at the lymph nodes eventually. So that's part of our defense mechanism. Now, we have some accessory lymph organs that are also somewhat closely or loosely associated with some of the tracts and ducts. We have the tonsils, and we'll look at the main tonsils. Those have an immune system function, and we have the vermiform appendix, sometimes just called the appendix, that sticks right off of the cecum of the large intestine. For a while, we thought that these tonsils and appendix didn't really have much function at all, but we know that they do have some immune system function, and so they are helpful, and we'll look at those as well. What
What does the bolt do?
So that is a lining of diffuse lymphoid tissue cells and lymph nodules, which help prevent the pathogens, let's say bacteria, from gaining access from our respiratory tract. So it's bronchial-associated lymphoid tissue. So instead of respiratory, it's bronchial is what they're calling it, but it's part of our respiratory defense system, so it can't get in. So if you're breathing something in and it's going down your trachea or something, it doesn't sneak in as easily across that mucosa because of that. So that's a little weird name and abbreviation, just like the GULT is the gut associated, is a little bit weird too, but that's what it's doing. Now, in addition to these structures, the bone marrow produces most of our immune system cells. All the white blood cells, all the red blood cells as well, but they're not part of the immune system. And all of them are made in the bone marrow, the red or active bone marrow. The thymus is active when we are young, and that's going to help in the differentiation and maturation of the T lymphocytes. We'll talk more about that. And so it's near the heart, and it involutes, it gets smaller as we age. Can be called, as we age, a senile thymus. The spleen, instead of having lymph fluid circulate, has the blood circulate through it. And it also has immune system function as well as removing old, worn out red blood cells, along with the liver also does that. So these are some of our major structures and organs that we're going to be looking at eventually. Well, what about the basics of the immune system? We have some capsulated lymphoid tissue. What does this mean? It just simply means they have a capsule around it. It's an organ that's part of the lymphoid response, and this includes the thymus, the spleen, the tonsils, and the lymph nodes. We're going to go over each of these histologically eventually. And then we have ones without a capsule. This is simply called diffuse or spread out lymphoid tissue. Here we're going to have lots and lots of lymphocytes. We also have macrophages there. When we look at the immune system, we tend to divide it into the innate and the acquired immune system. The innate and the adaptive or acquired is also called. The innate and adaptive or acquired immune response. These are both part of the immune system. They both work together, but they have some different characteristics, and so we divide it into these two major areas. First of all, the innate immune system is less specific. It's going to attack anything that looks foreign, but in a non-specific way. It has no immunological memory. That means if you've been attacked by a certain virus or bacterium, and you get attacked again by that same one later, the innate system doesn't remember, but the acquired system does. So there is a part of the immune system that has memory, but the innate system does not have memory. When we look at the innate immune system, we have lots of proteins and cells. And as we look at the entire immune system, we have lots of proteins and cells which help us fight off these infectious agents. And it can get a little bit complicated and overwhelming when we have so many different types. But if we think of it as, yes, we have all these different proteins and all these different cells that are helping us, and some of these cells are secreting some of the proteins, but some of the cells are also secreting other chemicals called cytokines. They can help attract other cells of the immune system to the area. They can help fight off the pathogen. they can help with an inflammatory reaction. So we have a number of different things there. When we're looking at this non-specific innate immunity, one of the protein types that we see is something called complement. Complement is a series of proteins that are found in the blood, and when these proteins get activated, they can help fight off infections in a non-specific but very important way. And it's just called complement. It's a protein. Another different category of proteins are called toll-like receptors. Receptors bind things based on their shape. And these toll-like receptors can bind different things and initiate immune system responses. For instance, toll-like receptor 4 recognizes lipopolysaccharides of the gram-negative bacteria. So once you get some of that connection, you'll start fighting bacteria. Toll-like receptor 2 recognizes the peptidoglycogen of gram-positive, peptidoglycan of gram-positive bacteria. So you can fight off gram-negative and gram-positive bacteria that way. Others fight against the viruses. And a lot of their ability to fight this off once you have a binding is to release chemicals, cytokines, like interleukins. This can also help lead to the adaptive or the acquired immune system being activated. So we also look at a lot of cells. Mast cells, which we looked at quite a bit, haven't we? They release a lot of chemicals, histamine and heparin and a bunch of others to help fight off infection. Eosinophils work predominantly against the parasites, you may remember. Neutrophils work predominantly against bacteria, they're phagocytic. Macrophages are phagocytic as well. And natural killer cells are a type of cell that also is going to be in that innate immune system. The adaptive immune system has 4 characteristics that the innate immune system does not have. Its ability to distinguish self from non-self, its memory, its specificity, and its diversity. All of these are important. And so, although it takes a longer time for the adaptive immune system to formulate its response, the response is larger, and it could be, so in some ways the innate immune system helps us not get the infection, but if we do get the infection, it tries to hold on until the adaptive immune system can gear up, and then that adaptive immune system can knock it out. In addition to these ways to classify, we can also say that we have a humoral immune response. That means in the body fluids, like the blood. Antibodies, for instance, the type of protein, defense protein, are going to be working in the blood predominantly. Other things are going to be more cell-mediated responses. This means it's going to attack cells which are altered. For instance, maybe they have a virus infecting them, or maybe they're a transplanted cell, or maybe they're a cell that is turned cancerous, tumor cells, and the like. So that's cell-mediated. Again, both work, the humoral and the cell-mediated. We divide them simply because they have a little bit of different place of action and target of action. This will show you, we went too far, phagocytosis, which you're familiar with, but it also illustrates some of the effective complement protein to help in phagocytosis in a process called opsonization. Let's just remind ourselves
components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism.
Just at the bottom, the red one.
Oh, thank you. Let's get rid of that.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So here's something that you're pretty familiar with. A lot of pathogenic organisms, like bacteria or something, can be phagocytized, and they can be fused with a lysosome, which has digestive enzymes, and we destroy it and say, hey, it's gone. And so that is very helpful. We have a lot of immune system cells which are phagocytic. Complement, which is this category of proteins in the blood, can help in this process by coating, let's say, the bacteria and making it easier to grab hold of by these phagocytic cells and get phagocytized. Well, sometimes, what about viruses? Viruses don't live on their own. They have to have a host cell. It could be our cells, it could be a plant cell, it could be invading plant cells. There's different kind of viruses that invade different kinds of cells, animal cells, plant cells. We also have viruses that infect bacterial cells. They're sometimes called bacteriophages or phages. These are viruses that infect bacteria. Well, if a virus comes into our host cell, it can take over our genetic material. It can sneak into our DNA. It can be an RNA virus, and it can have a reverse transcription and get into our DNA, or it can be a DNA virus. And what it can do is it can take over the genetic instructions of the cell and either sort of hang out there in a stealth-like fashion, or it can replicate and form a whole bunch of viruses and explode our host cell, and then the viruses go on and infect other cells. Well, one way that we can defend against the cells is if we have a host cell that's infected with a virus, even though that host cell cannot directly knock out that virus, it can produce a chemical cytokine called interferon. And that interferon can go to other of our host cells that have yet to be infected with a virus, and it preps them so that when the virus comes and attacks them, they are able to destroy it. So this is another way that the body defends itself, in this case against viruses.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
So that's another mechanism where the body defense system is trying to protect itself, protect the host. Now, when we look at the immune system, we're going to look quite a bit at two categories of lymphocytes. You remember lymphocytes? And those are T and B lymphocytes, and then we'll have some subcategories of each. And we'll also be looking at what are called antigen-presenting cells. Antigen-presenting cells are quite interesting and unique. They phagocytize a pathogenic organism like a bacteria or something, and they put a piece of that substance, bacteria or something, on their surface, and they present it to other cells and help activate those immune system cells, like T and B lymphocytes. Let's talk a little bit more about that. First, before we go to that next slide, we have a number of antigen-presenting cells, lots of macrophages, dendritic cells, Langerhans cells, M cells, B cells. These can all phagocytize bacteria and present a small piece of that to other cells and activate them. So how are we going to do this? Well, this is a diagram from your book Atlas, and it talks, it gives you a nice overview of how we activate T cells and B cells. T cells and B cells are lymphocytes, and they can exist as naive, unexposed, and then they can be activated to more mature, differentiated. When we look at this, we started out with a phagocytic event by an antigen-presenting cell. So here's our antigen-presenting cell up here. It phagocytizes something like, let's say, a bacterium or something, and it expresses a little piece of that antigen on its surface. And it does that on a particular type of receptor type. These are called major histocompatibility receptors. We have class 1 and class 2. Essentially all of our nucleated cells have major histocompatibility class 1 on their surface, and the antigen-presenting cells also have a second type, major histocompatibility class 2 receptor. So if we look at that where the little pointer is, there is the receptor type there. Now, when you take the little piece of the antigen and you put it right there on the major histocompatibility receptor, type 1 and type 2. It's sometimes described as a hot dog in a bun. The bun would be the receptor, the MHC receptor, and the hot dog would be the little piece of antigen that's being presented. Then along comes some T cells. Now, we can subclassify T lymphocytes if they have a receptor called CD8 on their surface. See the CD8 here? That's called a cytotoxic T helper cell. If they have a different receptor called CD4, that's called a T helper. Now, if a cytotoxic T cell with its CD8 receptor binds to this complex of MHC and presented antigen, and also a T helper cell binds with its CD4 receptor, the MHC2, and the presented antigen, the T cell becomes activated. It divides and becomes activated. The T helper cell helps in this process by releasing interleukins, interleukin 1 and 2. This causes the cytotoxic T cells to expand and form more of them. And eventually these these cytotoxic T cells, if you have one of your own host cells that's infected with, let's say, a virus or something, and it's been presented that antigen, that T cell will then recognize that defective, that infected host cell. It will release some chemicals and it will destroy that altered cell. They're called cytotoxic because they destroy our host cells that have been altered, have been infected. In addition, when the T helper cells get activated, they can come over and activate the B lymphocytes. The B lymphocytes, instead of having receptors on their surface like CD4 and CD8, they have antibodies on their surface. And antibodies have this shape, looks like a little Y. Antibodies, by the way, are also called immunoglobulin, same thing, right? So on the surface you have antibodies, and each B lymphocyte with antibodies on it will have a slightly different shape, a variable region on the antibody, and so it will bind to a particular different shape. If it gets the same antigen that got eaten up by the antigen presenting cell, it can bind to the T helper cell that was activated, again by releasing interleukin chemicals that will activate the B lymphocyte to what we call plasma cells. Histologically, we know that plasma cells are a little bigger and usually have an eccentric nucleus, but what do? The plasma cells produce lots and lots of the same antibody that was recognized on the original B lymphocyte. And those antibodies can do many, many things to kill the invading pathogen. They can punch holes in the wall of the pathogen, like the bacterium. They can make it easier to phagocytize, opsonization, etc. They can release chemicals. A lot of times, a subset of these B lymphocytes that are activated will become memory B cells. They'll be held in reserve, and if you get the same antigen presented at a later time, they can quickly become plasma cells and become activated. So that is sort of the overall scheme of how we activate T and B lymphocytes. Here's kind of an overview of it.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest